Good morning, and thank you for joining our March Science for Staff lecture. Please mute your phones and disable your video if you have not already done so. My name is Andrew Bowe, and I'm on the Hutch Connect and Science for Staff committees that bring you this series. Please visit the Science for Staff page on CenterNet for more information on the Science for Staff series. There you can view previous lectures and see our upcoming presentations listed. We have a unique presentation today in that we have 4 featured speakers this morning. For today's lecture, we're spotlighting the genetics and epidemiology of Colorectal Cancer Consortium, otherwise known as GECCO. Our 4 presenters are Dr. Riki Peters, Professor and Associate Director in Public Health Sciences, Tabitha Harrison, GECCO's Coordinating Center Manager, Dr. Jeroen Huyghe, Senior Staff Scientist in Public Health Sciences, and Dr. Lee Su, Professor in Public Health Sciences. Feel free to submit any questions that you have during the lecture. You can submit questions during this lecture with the chat function in Zoom. At the end of the seminar, I'll go through and read the questions to the group. Welcome, and thank you to the 4 of you for joining us this morning. Dr. Peters will be starting us off.
Thank you, Andrew, for the nice introduction. Yeah, we are excited to share some of our ongoing research on colorectal cancer with you. Our research is highly collaborative and requires a mix of different expertise, both within the Fred Hutch team as well as through numerous collaborations we have established across the globe. We have split the presentation in 3 areas led by different team members. First, Tabitha Harrison, a genetic epidemiologist, will show our work on tumor profiling, followed by Jeroen Huyghe, a statistical geneticist who will describe our many discoveries for genetic risk factors. And then Lee Su, a well-known biostatistician in our faculty, will talk about how we can use all these discoveries we have made to build comprehensive risk prediction models for precision medicine. The work we are describing today is built on the genetic and epidemiology of Colorectal Cancer Consortium, also called GECCO. This is a consortium we started more than 10 years ago. It's a still growing consortium, which includes investigators from more than 70 studies currently, and includes more than 150,000 participants. Our initial focus was on discovering genetic risk factors and gene-environment interactions. However, we have expanded substantially from there, including other areas like tumor profiling, survival, or risk prediction. I also like to mention a recently funded project that is building on collaboration started within GECCO and is specifically focusing on colorectal cancer health disparities. As you can see from the histogram on the left side, colorectal cancer rates vary dramatically by different racial ethnic groups and are highest in Alaskan Native, followed by African American people. We aim to develop a large program that is focused on health disparities and are currently in the process of building large resources. In our first projects we are funded for are focusing on gene expression, immune profiling, and the microbiome. Before we start, I would like to take the opportunity to say thank you to so many at the Fred Hutch for all the wonderful support we have received over the many years. While we likely have missed some, you can tell from the long list, it truly takes a village to do the type of research we are doing. We are very grateful for the strong and professional support we have received from you. This really makes all the difference for us. With that, I give the floor to Tabitha Harrison.
Thank you, Riki. So I'm going to talk about one of several areas of research within GECCO, and that is to look more specifically at colorectal cancer tumors and to understand how tumor features relate to colorectal cancer risk factors and disease outcomes. So next slide. Thanks, Riki. So first, I want to distinguish between research that we're doing to better understand the characteristics of people who get colorectal cancer. So thinking about a person's genetic or lifestyle risk factors versus where we're trying to better understand aspects of the tumors themselves. So when we talk about a somatic mutation, we're talking about alteration in DNA that occurs in the tissue after conception. So it's not something that would be inherited or passed on to children, but these alterations can cause cancer or other diseases, although not always. So we're looking both at genetics and tumor features of the tumor, and then we're also looking at genetics and features of the what we call quote unquote the host. Next slide.
So I'm not sure why I have problems moving forward. It will be probably a little delayed, sorry.
Okay, no worries. So part of our motivation for somatic research is driven by knowing that there are differences in tumors and that different types of tumors have different relationships with known risk factors. So we can think about the location of the tumor within a colon or other clinical features, and Jeroen will share with you some of our research in that area relating tumor location to germline genetics. But we also think about molecular features of tumors. So thinking about what types of mutations we see and how that may be indicative of how the tumor develops from normal tissue into cancer. But we're also learning on recent history that the immune response to tumor cells is really important to understanding how the tumor develops and thinking about subtype classification. So there's not yet a gold standard for thinking about tumor subtypes, but we know that once we can characterize and subclassify tumors, we can then get a better understanding of how these relate to known risk factors and survival outcomes. Next slide.
I really am not sure why I have such trouble. Sorry.
That's okay. We can just have a breather between each slide. So the other thing I want to mention is that we're really learning how important it is to not only understand the tumor itself, but to think about the cells and molecules and structures that surround tumor cells, or what we call the tumor microenvironment. And we're learning that constituents of the microenvironment play really important roles in terms of understanding development, progression, and therapeutic response. So for instance, we already know about specific mechanisms identified for specific bacteria or bacterial toxins in colorectal cancer. Bacteria like Fusobacterium nucleatum have been found to be significantly elevated in tumor tissues and stool specimens of cancer patients relative to those in normal controls. And we also see poorer outcomes with increased amounts of Fusobacterium nucleatum. We're also learning, again, as I said earlier, about the importance of understanding collective effects of the microbial community and inflammation. So we know that the inflammatory microenvironment is an essential component for tumors, where inflammation and changes in immune response play important roles in tumor initiation and development. Next slide. So what we're doing right now in GECCO is to get more detailed characterization of colorectal tumors on a large scale. So to do this, we amassed tumor molecular data that studies had already collected, commonly assessed features like mutations in BRAF or KRAS genes, where they did more detailed molecular characterization among a subset of 12,000 cases. And then in addition to getting these data, we wanted to more specifically get more detailed information about DNA mutations. And we did this in about 5,900 colorectal cancer cases and about 250 adenomas, which are the precursors to colorectal tumors. And we did this through targeted sequencing to better understand gene mutations. And now we're in the first year of a grant to also characterize immune response and specifically to look at T cell density and how these T cells are distributed relative to tumor cells. And for all of these tumor and microenvironment data, we're then relating them back to patient germline genetics, lifestyle factors, and survival outcomes. Next slide. So what we're learning this far is that cases that have colorectal cancer diagnosed before age 50 years of age are more likely to have certain molecular features. And these features suggest that the cancer is occurring through what we call an adenoma carcinoma sequence. And the main implication of that is that for colonoscopy screening will likely be successful in finding most of these tumors versus some tumors that have more flat lesions and may be more difficult to detect. We're also learning that heavy smokers have certain tumor profiles, and those profiles suggest that the cancer may be occurring through a different type of pathway or a serrated pathway. We also are learning that there's a common genetic variant that people, and germline genetic variant that may be associated and for people at higher risk for having a certain type of mutation profile, which is called a microsatellite instability. And that this relationship is greater for women and for tumors that are located proximally in the colon. Additionally, we're also learning that because of our sample sizes of these studies, we can start to look at subspecies. So not only that Fusobacterium nucleatum is associated with colorectal cancer survival, but also specifically the subspecies. So these are just examples of some of the types of things that we're learning from this research, but we're also learning things like different subtypes may matter for our combination of molecular markers may matter for survival time. Next slide. And as Riki mentioned earlier on, this research is possible because of Fred Hutch staff. And I just also want to point out that the research is possible thanks to our many international collaborators in GECCO who've contributed data, their varied expertise in areas of research, and also by taking on a lot of these analyses that I've described. Next slide. So I want to present today also on an area where we're addressing a critical gap in the somatic research that we're doing. And as Riki showed earlier, there's a striking disparity in colorectal cancer incidence and mortality among Alaska Native people, and an elevated risk for African Americans when compared to non-Hispanic white and Latino populations. So most of the somatic research that we've done to date, including our own and what I showed you, has been done in samples from non-Hispanic white populations. So there's no somatic research studies for Alaska Native cases and very few for African American cases. So we don't know what genes and microbiota might be contributing to a person's risk of dying from colorectal cancer in these populations. Next slide. So we started the translational research program in cancer disparities, and we're doing now some molecular characterization of tumors in Alaska Native, African American, Latinx, and non-Hispanic white populations. And the primary goals of these research is to look at gene expression in tumors, and also by doing characterization to identify the presence and abundance of different bacterial species through droplet digital PCR and sequencing. And we're also going to build the infrastructure to do future studies. And I just want to highlight that you'll recognize some of the Fred Hutch folks involved in this work. And we're also partnering with the Alaska Native Tribal Health Consortium, the Oshner Healthcare Systems in Louisiana, and Cedars-Sinai in California. Next slide. I won't go into details here, other than to highlight that as part of this project, we have a core for processing tissue blocks, 2 main research projects, which I'll touch on in a second, and then a pilot project program as well. And then we have internal, external, and community advisory boards guiding this work. And we also have a key team, the executive committee leading the program. Next slide. So for biospecimen processing, we're receiving blocks in-house at Fred Hutch. And again, this is where we're very reliant on the infrastructure and the resources that the Fred Hutch is able to support this research. So we're doing pathology review and taking digital images. We're also collecting clinical and medical records data for analyses. And these will be utilized both for the main research projects and also for future grants and pilot studies. Next slide. So without going into too much detail, I just wanted to highlight the 2 main research projects, and mostly in terms of what we hope will be the impact of this research. So I want to mention the main goal of the first project, the Gene Expression Project, is to reduce longstanding disparities in colorectal cancer outcomes by developing clinically useful predictors of cancer outcomes that can guide clinical decision-making and potentially identify therapeutic targets. And next slide. And then the second project, to characterize bacterial presence, is to help really inform strategies for targeted treatment for things like using antibiotic or other bacterial species, or to develop interventions such as dietary changes to prevent disease and improve health outcomes. So this is an exciting area of research for us, and a lot of these things that I described, we're just starting to get them off the ground. And I think it's, again, I just want to reiterate what Riki had said. A lot of this is possible because of the partnerships that we have, both within the Hutch and within the consortium. So with that, I think I'm going to pass it over to Jeroen.
Thank you. I have one question to Andrew before Jeroen is starting. Am I the only one who can admit people who are waiting in the waiting room? I'm happy to do this, but I figured out this is the reason why I had problem moving slides forward, but now I can do it.
Oh, I see. No, we can take care of that as well on the back end, so you don't
Okay,
need to worry about that.
you see them too.
Yep.
Thank you.
No problem.
Jeroen.
All right, good morning, everyone. So I'm going to talk about our work focused on the discovery of inherited genetic risk variants, which are also called germline or host genetic risk variants. Next slide, please. So colorectal cancer is a complex disease with many different genetic, environmental, and modifiable lifestyle risk factors contributing to disease risk. And those that accumulate a lot of risk factors are at particularly high risk. And modifiable risk factors, they include consumption of processed and red meat and habits associated with an unhealthy lifestyle, such as smoking, drinking, low intake of vegetables and fruit, and a sedentary lifestyle. And modifiable protective factors include habits associated with a healthy lifestyle, such as physical activity, factors related to eating a healthy diet, but also certain drugs, use of certain drugs such as aspirin. And in contrast to lifestyle factors, genetic risk factors are not modifiable. So we know that a small fraction of patients is affected by a well-defined hereditary syndrome caused by a defect in one of about 18 genes. However, the majority of patients are sporadic colorectal cancer cases where genetic risk is determined by the cumulative effect of a large number of genetic variants. Next slide, please. So before talking genetics, I wanted to show this slide. So we looked at associations for risk and protective factors in the GECCO Consortium. And for this, we harmonized epidemiological data for over 60 studies. And so as expected, we find highly significant associations for many risk factors like processed and red meat consumption, obesity and smoking, and several protective factors like some dietary factors and drugs. Next slide, please. So how much of the variation in cancer risk between individuals can be explained by genetic factors and by environmental factors, including lifestyle factors? So in other words, what are the relative contributions to risk? So historically, the genetic contribution, which is called heritability, has been estimated using twin studies. And based on the largest twin registry study, the heritability of colorectal cancer is estimated to be around 35%. However, it's important to realize that this estimate of the genetic contribution does not tell us anything about what this looks like in terms of number of genetic risk variants, their effect sizes and their frequencies in the populations, or in other words, what is called the genetic architecture of the disease. And until recently, we essentially knew nothing about this. Next slide, please. So genetic studies of complex diseases, including sporadic colorectal cancer, really lifted off only in 2007, when genome-wide association studies, or GWAS, became available. So before that, there had been many unsuccessful attempts to identify risk variants contributing to colorectal cancer. So in the early 2000s, when many so-called candidate gene studies were conducted, these were unsuccessful because we didn't really know where to look in the genome, and also studies were too small and therefore statistically underpowered to detect these small effect sizes. And GWAS became really possible due to breakthroughs in three different areas. So first, the availability of high throughput genotyping technologies, in particular genotyping microarrays, and also next generation sequencing. And second, advances in our understanding of the human genome and DNA sequence variation in human populations, initially enabled by the Human Genome Project, which was completed early in the century, and then by projects such as the International HapMap Project and the 1000 Genomes Project. And these projects were driven by advances in next generation DNA sequencing technologies and also rapidly dropping sequencing costs. And last but not least, advances in statistics, in particular insights in study design, and also in genotype imputation methods that predicts genotypes for variants not included on microarrays, which allows combining different GWAS datasets and studies in a meta-analysis. Next slide. So as an example, here's a summary of a GWAS for colorectal cancer that we published a couple of years ago, and that was the world's largest GWAS for colorectal cancer at the time. And the figure shown is a so-called Manhattan plot, which shows the statistical association test results for all 17 million variants we tested. So each dot is the p-value for one's variant. And on the y-axis, the minus log p-value, which is essentially measuring the signal strength, is shown. And on the x-axis is a chromosomal position. So in this study, we detected 40 new risk variants, which are indicated by the red signals. And most variants discovered in GWAS are common in the population by design, but we also discovered the first rare variant for sporadic colorectal cancer and several low-frequency variants. And importantly, this GWAS also provided new leads for by identifying several genes in pathways not previously implicated in colorectal cancer risk. And this is actually one of the main reasons why we are interested in discovering new genetic risk loci. So we want to advance our understanding of disease biology, which can inform drug targets to improve treatment of disease and also for chemo prevention. Next slide. So this year, 2021, actually marks the 15th anniversary of GWAS. So this figure shows the cumulative number of colorectal cancer risk variants discovered since 2007. So before 2007, no variants were known. And initially, the number of variants increased rather incrementally when sample sizes were only in the thousands. And the inset shows how GWAS sample sizes increased over the years, and also that very large sample sizes are critical for additional discovery. And the latest wave of GWAS includes data for close to 250,000 participants now, and we do not see saturation yet in terms of number of new variants discovered. Next slide. So what do we know today about genetic risk factors for colorectal cancer? So we know that 5 to 7% of patients are affected by well-defined hereditary syndromes caused by genetic defects in one of 18 genes. So many of these genes, they were already discovered in the 90s and the 2000s by family-based linkage studies using large extended pedigrees and positional cloning. And some of these syndromes are the most frequent ones are Lynch syndrome, familial adenomatous polyposis, and U2IH associated polyposis. And GWAS discovered 180 mostly common genetic risk variants. And I want to point out that we contributed to the discovery of over half of these risk variants. So however, 2/3 of these genetic risk factors, they remain unidentified. So it's estimated that there's about an additional 1,500 unknown common variants that await discovery. And there's also an unknown number of rare variants. Next slide. So this mathematical modeling study inferred that there's about 1,500 independent common risk variants that are still unidentified. And it suggests that we need close to 400,000 samples to identify 80% of these. Next slide. So what are the next steps? So first of all, GWAS conducted so far, they have been predominantly based on non-Hispanic white participants. And there's, of course, an urgent and critical need to increase racial ethnic diversity in GWAS to avoid the risk of exacerbating health disparities. And then, as shown on the previous slides, sample size and large-scale international collaboration across GWAS consortia are critical for new discovery. However, substantially increasing sample size is becoming increasingly difficult. Next slide. Therefore, more targeted strategies to prioritize variants for testing are an active area of research, and these includes filtering of genetic variants based on colorectal cancer relevant functional annotations. For example, epigenomic data tell you which genomic regions are active in the relevant cell types, and transcriptomic data can tell you which genes are expressed in the relevant cell types. Also, instead of testing single variants, we can group variants. For example, when they're located in the same gene, and we can use more powerful statistical methods to discover associations with sets of rare variants rather than doing single variants testing. A particularly promising set of approaches includes various machine learning approaches. So these include like testing association between genetically predicted gene expression or methylation and colorectal cancer risk. So these are called like transcriptome-wide association studies or methylation or methylome-wide association studies. And so several of, well, a few of these have now been conducted for colorectal cancer and yielded additional associations. And prediction of variant function using deep learning applied to high quality multi-omics data is also a very promising avenue. Additionally, analysis of more homogeneous patient subgroups can lead to new discoveries. So we have just published a GWAS based on case subgroups defined by anatomical sublocation of the primary tumor, which yields an additional 13 new risk variants. And other efforts are underway to look at subgroups based on tumor molecular subtypes, age of onset, and histopathological features. Next slide. Finally, discovery of rare variants is lagging a little bit behind, and is kind of currently unknown how many of these contribute to risk. So because GWAS is limited to identifying more frequent variants, and to in order to identify these, we need to use a different experiment, namely like large-scale whole genome sequencing studies. And currently, these are still very expensive to carry out in large numbers of subjects. Also, genotyping imputation plays a very important role in the GWAS study design. So we genotype samples using microarrays, which contain typically 500,000 to 1 million variants. And then we use large reference panels of whole genome sequences to impute, or which is predict, other variants in the genome, which is a highly accurate approach. And finally, experimental follow-up studies to identify the actual causal variants underlying the risk association signals we identified, and the target genes and understand the biological mechanisms are lagging behind, mostly because most of the variants have just been discovered in the past few years. And this is really necessary to realize the full translational potential. And promising in this regard are new high throughput technologies such as MPRAs, or massively parallel reporter assays, or CRISPR-Cas9 gene editing techniques that some of our collaborators are implementing. And I will end here and leave it to Lee.
Let me first unmute myself. So in addition to what Jeroen has described, the potential of genome-wide association studies, another important usage of this data is to predict risk for developing colorectal cancer. And this risk prediction model can be used for early detection and cancer prevention. Next slide, please. So why is it important that we detect cancer early? As for any cancer, the earlier we can detect it, the better is the survival rate. So as can be seen here, the 5-year survival rate for stage 1 cancer is 75%. And that survival rate reduces to 6% if the cancer is detected at stage 4. Next slide. So for colorectal cancer, we have very good early detection and prevention methods, such as colonoscopy or FIT test to test the blood in the stool. However, about 1/3 of the eligible people do not receive colorectal cancer screening. And there are several reasons for it. For example, the cost reason or inconvenience or just lack of awareness, among others. And the decision to determine who is eligible for screening is actually based on pretty simple criteria, based on age and family history. And the current screening guidelines state that everyone without a family history should start screening at age 50. Next slide. So at age 50, the average risk for developing colorectal cancer in the next 10 years is close to 1%. However, this is only an average risk, and there's a wide distributions. What that means is that some individuals may have a much lower risk, while others have a higher risk. So if we can identify those at high risk, then we can get them to screen early or more frequent. And that's exactly what we're trying to do with developing a risk prediction model. Next slide. So as shown in Jeroen's presentations, many genetic variants have been discovered to be associated with the colorectal cancer. And many lifestyle and environmental risk factors are also contribute to colorectal cancer development. So the question is how to summarize these risk factors. Perhaps most straightforward approach is to take the sum of these risk factors, and with each risk factor weighted by its effect on the risk of developing disease. So the effect can go both directions. For example, smoking increases risk, so the effect is positive. And the exercise is a protective effect and reduces risk for developing colorectal cancer. The effect is negative. So by summing all those risk and protective factors with appropriate effect size, we would have an overall assessment of risk. Next slide. So for genetic variants, we can sum the contributions of many genetic variants throughout the genome. And this score is called polygenic risk score. And sometimes it's also called genetic risk score or genome-wide score. It sort of reflects an overall individual's genetic predisposition for developing the disease. And it can be used as a predictor for predicting that disease. Next slide, please. Similarly, we can also sum the contribution of lifestyle risk factors, for example, smoking, alcohol consumption, obesity, and some dietary factors. And we call this environmental risk score. Next slide. So in the last few years, we have been working very hard on developing risk prediction models using genome-wide genetic data. So in order to build a risk prediction model with millions of genetic variants, typically we will have a data set for training, that is to identify genetic variants that are predictive of disease, and then use an independent data set to validate the model. And due to the modest effect of each genetic variant, typically we will need a very large data set so that we can reliably identify which variants are associated with the colorectal cancer risk, and to what extent the impact of these variants on the disease risk. So as can be seen here, the training data set includes approximately 120,000 cases and controls. And the validation data set is based in the Kaiser Permanente, Northern California, and that includes 100,000 participants. Next slide. So this risk prediction model work is led by postdoc fellow Minta Thomas. So she explored two main approaches for building the risk prediction model. So as I mentioned earlier, so we have millions of variants. So perhaps the most sensible approach is to first reduce the number of the variants to a manageable amount. So the first approach essentially is to first prune out the correlated SNPs that perhaps have the redundant information and select the variants that are most associated with the disease risk. And when the number comes to a more manageable number, then we can build a risk prediction model using machine learning algorithm. So the downside of this approach is that once the SNPs are pruned out, they will never be selected in the second stage of risk prediction model. So another approach that Minta explored is to not to prune. So instead of dealing with this giant matrix that includes 120,000 individuals and millions of variants, so she used an approach that based on the summary statistics, and that can reduce both computational time and storage for building the risk prediction model. So it turns out that actually the second approach that used all the variants, over 1.5 million variants, that predict the risk the best. Next slide, please. So this figure shows the probability of a developing colorectal cancer stratified by polygenic risk score. The average lifetime risk is just under 5%, but for individuals in the top 10% of polygenic risk score, the lifetime risk is over 10%. Whereas for individuals in the bottom 10%, the lifetime risk is below 1%. So the hazard ratio for being in the top 10% compared to the rest is 2.5 folds. To give you a general idea, having a positive family history of a colorectal cancer increases risk about 2.2 folds. Next slide. So genetic risk factors are not modifiable, as are some of other risk factors. For example, sex, age, and family history. However, some risk factors are modifiable, such as dietary variables, smoking, and alcohol. So the former postdoc fellows, Margaret Du and Wendy Wang, and they demonstrate that having a higher environmental risk score and a higher non-modifiable risk score is more than the sum of the risk of each risk score. On the other hand, it also means that if one is at high risk due to non-modified risk score, as shown in here on the Q4 on the rightmost of the figure. So by changing to a healthier lifestyle can potentially lower the risk for developing colorectal cancer substantially. So in the figure shown here on the Q4, which is the highest quartile of non-modified risk score, by changing the environmental risk score from the highest quartile to the lowest quartile, they reduce the risk to even below the average risk. Next slide. So another thought of using the risk score is to see whether we can recommend age to start screening based on the risk score. So this is a figure of recommended starting age for screening according to risk score. For positive family history that is shown on the left-hand side, and the negative family history that is shown on the right-hand side. So the horizontal lines are the current recommended starting age for these two groups. So if you use the average 10-year risk, 1%, as I showed earlier, for a 50-year-old as the threshold, we can see that some individuals may not need to start screening at age 40, even they have positive family history, as shown by the curve that's above the horizontal line on the left-hand side. On the other hand, some individuals who don't have positive family history may need to start screening earlier, as shown by the curve below the horizontal line on the right-hand side. Next slide. So there are limitations to our study. A major limitation is that most of our GWAS studies are of European ancestry. So the polygenic risk score that derived using the European ancestry data do not perform well in the minorities. And the reasons are that these genetic variants are markers. So they're tagging the causal variants. So because of the genetics architectures differs between different populations, so the genetic variants that derived based on the European ancestry data do not tag or capture the effects of the genetic variants in minority samples. Next slide. So we are in the process to get a large number of racially and ethnically diverse cases and controls. So this work is recently funded by the NCI. So we, under this grant application, we plan to develop and validate a polygenic risk score for colorectal cancer across all major racial and ethnic groups in the US. We will hopefully to get up to 28,000 African-American cases and controls, approximately 100,000 Asians, and 30,000 Latinx. Next slide. So we are also collaborating with the Kaiser Permanente Colorado, Andrea Burnett Harmon, whom many of you may know, as well as colleagues Charles Kuberberg and Linda Ko, to see whether we can evaluate the risk prediction models in a pragmatic trial. So in this proposal, we propose to randomize the participants who have not had screening by age 51 into two arms. One is just a usual arm to send them the usual care reminder, and the other arm is to offer this participant the genetic testing. So the idea is that participants who receive genetic testing and their results were more likely to initiate the screening than those who received the usual care reminder. I also want to add, actually, not just the genetic testing, we're also going to bring the risk assessment that based on the lifestyle and environmental risk factors. And we believe that participants with a higher than average risk based on the risk assessment will be more likely than participants with the average risk to initiate the screening and perhaps to choose colonoscopy. And this has been shown with a positive family history. The participant with a positive family history will more likely to take the screening. And we will also, of course, like to understand or how this risk assessment information shapes the participant or individual's perceived risk for developing colorectal cancer and the beliefs about the screening and associated anxiety. Next slide. Okay, so I'll pass this slide back to Riki.
Yeah, I just have 2 slides at the end to show, our team who's working at the Fred Hutch on this for many years. As I said, we started more than 10 years ago. And here's some pictures of our outings, close together without masks. I know, can't imagine this right now. And this is a picture of our investigator meeting that happened. The last one that we had in person was 2020. And this year, of course, we had a virtual meeting, which went really well, given the circumstances. But our investigators really enjoy coming to Seattle, even if it's in the winter. And a big thank you for everybody helping us to make this event happening. With that, should I stop sharing my screen for questions?
Sure, or you can just leave this last slide up. Well, let's see. I don't see any questions in the chat. We'll give people a couple of minutes, but in the meantime, thank you very much, Riki and Jeroen and Tabitha and Lee, for presenting about GECCO and the consortium. It's quite a sophisticated group of individuals and work that you've all linked together.
Probably you can use the time to give a reminder for everybody who's getting to the age of colorectal cancer screening. This month is the Colorectal Cancer Awareness Month. So it's really important to do the screening. We are not there yet to identify those at high risk, aside from the research settings. So this is something that will come in the future, hopefully to guide people